In this tutorial, we will show how to find a protein record in the Genophore database. Type in the search field the name of the protein or gene of your interest. Genophore will search across the entire platform for all records that are related to the search term. In this case, we'll look for the human PDCD1 receptor, which can be easily found under the UniProt tab. Click on the record named PDCD1. In the protein record main page, Genophore displays data related to the molecular organization and structure of the protein. One can readily tell from the displayed topology drawing that PDCD1 is a single-pass membrane protein. On the left-hand side of the protein topology drawing, you can find information on both the 2- and 3-dimensional structures available for PDCD1 within the tabs. The tab marked as 2D displays an interactive drawing of the secondary structure of the protein, extracted from experimental structural information. Genophore can also allow prediction of the secondary structure using algorithms such as the Garnier method. The 3D tab shows 3-dimensional atomic models of the protein, retrieved from the current protein data bank records. By default, a ribbon model will be drawn with post-translational modifications such as disulfides are highlighted in red. On the left-hand side on the protein structure visualization panel, you can browse all the currently available PDB records. Now back to the membrane protein topology visualization deck. From the protein molecular features panel, you can visualize a variety of different features on all of the structural schematics as well as on the amino acid sequence level. To search for a particular molecular feature, click on the Features tab and search for your feature of interest. Here we'll search for Ig-like domain. Checkmark the domain feature. Now Genophore will highlight the protein feature corresponding to the Ig-like domain in all of the 3 structural representations, including in the secondary and tertiary structural models as shown here in purple highlights on the beta sheets of the Ig-like domain. Genophore also automatically aligns the sequence of the PDB record to the native sequence of the protein. You can see here that Genophore flags in red highlights any mismatch between the PDB sequence and the native sequence, while all the identical amino acids are highlighted in green. Genophore automatically populates sequences of top orthologs to the human PDCD1 gene. You can perform a global alignment across all of sequences and subsequently sort them according to their similarity or identity. In order to view the available drugs targeting this receptor, click on the Drugs tab. You can see all the approved and experimental drugs that have been reported. You can also sort them according to the approval stage or to the disease indication. Here we will click on the biologic antibody drug pembrolizumab. This takes you to a page dedicated to this biologic with many important information and features. These features include the sequence of the antibody and the annotation of various CDR regions in both the light and heavy chains. In this antibody they are annotated according to the Cabot numbering, which is one of 6 numbering schemes made available by the Genophore platform.